Great, thank you very much, Eric, and thank you to PPMD for giving us this opportunity to share our exciting results from study 041 with Translarna or Ataluren. PTC Therapeutics was founded almost a quarter of a century ago with the mission of using pioneering science to develop important therapies for patients with rare disorders. Specifically, the initial focus was on developing drugs for boys with Duchenne muscular dystrophy. Our initial efforts focused on identifying oral nonsense suppression molecules, and the result of those research efforts was Ataluren or Translarna. When we began the Translarna drug development program, there were no previous drug development programs for Duchenne muscular dystrophy. That raised a number of challenges, in that we had to both learn how to study a drug for nonsense mutation DMD while also learning about the effects of Translarna at the same time. This included making important learnings about natural history of disease, patient populations, and appropriate endpoints to capture treatment effects over the course of a clinical trial. We were very proud in 2014 to receive conditional marketing authorization in Europe for Translarna, making it the first approved therapy for Duchenne muscular dystrophy. Translarna is now available commercially in over 50 countries worldwide, and we have really been on this journey that's had many setbacks, but importantly, many more successes that have allowed us to bring this therapy to patients. We're also incredibly proud of our partnership with the DMD community and are incredibly grateful to the patients, families, PPMD, other foundations, and our investigators who've really been along with us on this journey and proving that when you're going to do something difficult and you're going to do something pioneering, it really takes a community working together to do that. As we all know, Duchenne muscular dystrophy pathology results from mutations in the dystrophin gene resulting in a lack of dystrophin. This lack of full-length dystrophin makes muscle cells very sensitive to contraction and force, and over time with forceful muscle contractions, there's cell membrane injury, cell death, and ultimately muscle loss, which is then replaced by fatty and connective tissue. The concept of replacing dystrophin early in the course of disease is that by providing full-length dystrophin, we could decrease the sensitivity of cells to injury and preserve muscle and function. Dystrophin is produced through translation of the mRNA to form a full-length dystrophin protein, and Translarna basically enables the production of that full-length functional dystrophin in the case of nonsense mutations. When you have a nonsense mutation, there's a sequence in the mRNA that basically puts up a stop sign and stops the production of full-length dystrophin. Translarna is able to trick the cellular machinery to read through that stop sign and continue the translation process to form a full-length dystrophin protein. Translarna has been studied in a number of clinical trials which have demonstrated benefit on 6-minute walk distance, time function tests, and other important outcomes of disease. Shown here are the results from Study 7, Study 20, and importantly, the meta-analysis of both studies, which showed significant benefit of Translarna over placebo across all of these important disease endpoints. In addition to the data that we have collected in clinical trials, we've also been able to capture real-world evidence of Translarna benefit through our STRIDE registry, which is our post-marketing registry, which we have been conducting worldwide. When we conduct clinical trials, we really try to learn in a finite, defined period of time how a drug might actually work over the course of a disease and in the real world. In other words, outside of that environment of a clinical trial. And therefore, what we're able to do with the STRIDE registry is collect these long-term data that can provide important information on the benefits of Translarna, in particular with regards to key aspects of transition periods of the disease, such as loss of ambulation and loss of pulmonary function. And as of the most recent data cut in 2021, we have data from boys in 13 countries at 64 different sites and 288 patients that can provide these important data on the real-world benefits of Translarna. Importantly, we've been able to demonstrate using a matched control from the Synergy database that Translarna can preserve ambulation. We have reported a delay of 5.4 years in the loss of ambulation, obviously very meaningful over the course of DMD. We have similarly been able to record important impact on preservation of pulmonary function with a 1.8-year delay in the loss of pulmonary function. So these results provide evidence in the real world over a longer period of time that Translarna is delivering important benefits over the course of the disease on two key morbid aspects of disease: loss of ambulation and loss of pulmonary function. These data from STRIDE, along with the safety data we have collected over the years, have supported the annual renewal of the conditional marketing authorization of Translarna in the European Union. We most recently reported a positive opinion for the 8th consecutive renewal of Translarna's authorization in Europe. Another important commitment as part of the conditional marketing authorization in Europe is the conduct of a placebo-controlled trial to confirm the favorable benefit-risk ratio of Translarna in nonsense mutation DMD boys. This is our study 41, and I'll now move to explain to you a little bit of detail about the trial and share the exciting results. Study 041 is a 144-week trial that included a 72-week placebo-controlled phase during which patients would receive either placebo or Translarna, followed by a 72-week open-label treatment phase in which all participants received Translarna. The key eligibility criteria were age over 5 years old, the presence of a nonsense mutation, and a stable steroid regimen, as well as a baseline 6-minute walk distance of greater than 150 meters. The key outcome measures were the 6-minute walk test, the North Star assessment, and time function tests. Patients were stratified based on a number of key characteristics, including baseline walk distance, specific type of steroid used at baseline, as well as time from supine to stand. So this was a broad population studied over the course of 144 weeks, and today I'm sharing the results from that 72-week placebo-controlled phase. This table summarizes the overall intent to treat, or ITT, study population. The first thing you'll notice is this is a very large study population, 359 boys, which is the largest prospective study done in Duchenne muscular dystrophy. The take-home point on this slide is that the two groups, Translarna and placebo, were balanced. They looked the same in terms of age, 6-minute walk distance, type of steroid use, and time from supine to stand. So the populations are very similar, and therefore we can make comparisons between the treatment effect and placebo. Next, I want to highlight the study disposition. This tells us, importantly, that the vast majority, over 97% of patients who entered the study, got to that 72-week evaluation. That's really, really important in a clinical trial. And I would say that in any clinical trial that can have 97+% of participants complete the final assessment is really incredible. But when you consider that the study was conducted during COVID, where we had challenges at our global sites, this is really, truly remarkable. And again, allows us to say that this study was very well designed and well conducted, and therefore the results have very important meaning. So now the results. First, importantly, we demonstrated, really for the first time in an ITT population in a prospective clinical trial, a statistically significant benefit of therapy over placebo. We see on the 6-minute walk distance a statistically significant benefit, and for Europe, the primary endpoint was defined as the rate of change, or the number of meters lost per week, and we see that therapy with Translarna slowed the progression of loss of 6-minute walk distance over 20%. That's really a meaningful and statistically significant finding. In addition, the time to loss of 10% of baseline 6-minute walk distance was also statistically significant and favoring Translarna, which slowed the achievement of that milestone by 50%. And Craig McDonald spoke yesterday about the clinical meaningfulness of endpoints, and he has written in the past about the importance of this 10% loss of baseline walk distance as being predictive of ultimate loss of ambulation. And so these data that we're seeing here in terms of slowing of loss of 10% 6-meter walk distance are really consistent with what we've been reporting in the STRIDE registry, which is that long-term treatment with Translarna slows the loss or delays the loss of ambulation. And finally, we see that almost twice as many boys in the placebo group lost ambulation during the course of the 72 weeks as compared with the Translarna treatment group. Next on North Star, we again see a statistically significant benefit of Translarna on both the total North Star score as well as the linearized score. And again, we're seeing about a 20% improvement in scores in the treatment group relative to placebo. And finally, on time function test, we again see on the 10-meter walk and the stair ascend a statistically significant benefit of Translarna over placebo. So overall, very impressive results, and we're incredibly proud to be able to present these really unique data showing a statistically significant benefit of a therapy over placebo in an overall intent to treat population. And importantly, these data do align with the real-world benefits we have been recording in the STRIDE registry. This chart digs a little bit deeper into the North Star scores and provides a little bit more granular information on the clinical meaningful benefit of that statistically significant difference between Translarna treatment and placebo. What we have here is 16 of the items from North Star, and we're comparing the proportion of patients who lose the ability to achieve these different activities, either going from 2 to 0 or 1 to 0 in treatment with Translarna versus placebo. And what we see is on virtually all of the different items in North Star, there is a protective effect of Translarna treatment in terms of loss of the ability to perform these different tasks, including in some cases a reduction of 30 to 40% in the risk of losing the ability to perform one of these tasks. So again, more evidence of clinically meaningful benefit of Translarna therapy. I showed you this slide earlier, which reported the results in the overall population from our previous placebo-controlled studies, Study 7 and Study 20. In reviewing the data from those studies, we also looked very carefully at trying to identify responder populations or specific subgroups who are more likely to demonstrate treatment effect over the course of the trial. This is something that was very important, we believe, to do because we actually didn't believe that it would be possible to demonstrate a statistically significant benefit in an overall population given the heterogeneity of Duchenne muscular dystrophy. And so what we learned is the subgroup of boys who had a baseline walk distance of 300 to 400 meters really were much more sensitive to treatment effect over the course of a clinical trial. And this made a lot of sense considering that boys who entered the studies under 300 meters at baseline went on to lose ambulation so quickly that it was really very difficult to be able to demonstrate benefit in walk distance over the course of a trial. And boys who had a baseline walk distance over 400 meters were stable. In other words, they weren't losing much walk distance over the course of the trial, which made it really, really hard to capture a treatment benefit. And so what you see on this slide, first on the 300 to 400 graph, is a summary of the difference in walk distance between treatment and placebo, confirming that we had a very significant effect, protective effect in the 300 to 400 group. We did additional work using artificial intelligence to say, could we find a more specific predictor that would further increase our chance of being able to capture treatment effect over the course of a placebo-controlled trial. And that brought us to the 300, so baseline walk distance of greater than 300 and supine to stand of greater than 5 seconds. And so based on these analyses, we decided to make that group our primary population. Again, not believing that we'd be able to demonstrate the effects that we saw in the ITT population. So let's look and see what these subgroups look like in the trial. So first, the 300 to 400 group. Again, very similar characteristics in terms of age at time of start of the trial, very similar in terms of walk distances, steroid, type of steroid used, and supine to stand time. And this is the 300 and greater than 5 group. Again, very similar in terms of key characteristics for the Translarna group and the placebo group. When we look at the results, we see again in the 300 to 400 group, not only the statistically significant benefit of Translarna over placebo that we saw in the overall population, but we see the differences are greater. So here we're seeing a difference in walk distance of 24.2 compared to the 14 meters we saw in the overall population, confirming once again that this is the population in a clinical trial that is most sensitive to treatment effect. So this is statistically significant on the walk distance, on North Star and time function tests, as we saw in the overall ITT population, but the differences between the two groups are greater, exactly as we expected. Now, unfortunately, in the 300 and greater than 5 group, we did not see a greater treatment effect. We did see benefit on all the endpoints of Translarna relative to placebo, but the differences were not much greater like we saw with the 300 to 400 group. I also want to provide some data we have on the upper limb test. This is something that obviously is particularly important as boys transition to non-ambulatory stages of disease. And we saw, I'll first point out there were very few patients over the course of this study who demonstrated changes in upper limb function, but we were able to demonstrate in those who did a benefit of treatment in the overall population and a statistically significant benefit of Translarna treatment on the performance of upper limb test in that 300 to 400 meter subgroup. And then finally, we performed a pooled analysis. We took the results from study 041 from the first 48 weeks and pooled them with the results from study 7 and study 20, which were 48 weeks in length, which is why we only used the 48-week data from study 41. And what we see here is a highly statistically significant benefit of Translarna treatment over placebo in over 700 boys. So these data are really demonstrating over this entire population of boys studied in our placebo-controlled trials, a highly statistically significant and meaningful impact of treatment on the disease. I'll point out that we did the same analysis, pooled analysis with the 300 to 400 subgroup, also highly statistically significant, and also, as I've talked about, a greater difference between treatment and placebo, confirming again the responsiveness or the sensitivity of this population to treatment effect. Importantly, in terms of safety, Translarna was well tolerated without any drug-related serious adverse events over the course of the trial. There were no adverse events leading to study discontinuation, and the number of adverse events between treatment and placebo were similar. So overall, strong signal of safety and tolerance of Translarna over the course of the study, which is consistent with the safety data collected across all of our previous studies. So in summary, for the first time, we've been able to demonstrate for a disease-modifying therapy a statistically significant benefit of treatment relative to placebo in an overall population. And this evidence from this trial really adds to the totality of evidence that we've gathered from our previous clinical trials, as well as from our real-world STRIDE registry. And again, Translarna demonstrated a favorable safety profile, which is consistent now with the safety data we've collected in over 3,000 patients treated with Translarna. The statistical analyses that we use for the data I just shared were done using what's known as a mixed effects model, which is a very robust way of analyzing data in a clinical trial when you make many measurements for each boy. So these boys underwent assessments at baseline and then again at 12 weeks, 24 weeks, multiple times over the course of the trial up to that last assessment at week 72. We were asked by the FDA to also analyze these data using something called an ANCOVA, which basically simply says, just look at the last observation at week 72 and compare that to the baseline observation and tell us the difference. So a really less robust way of looking at data, but one important nonetheless for understanding how well that mixed effects model estimated the differences between placebo and Translarna. And what we see in the first column has the MMRM, or the mixed model data that I showed earlier, the ANCOVA data on the right. And what we see is the results are very similar. The differences between treatment and placebo are very similar, not surprisingly, given that the ANCOVA doesn't provide as robust an analysis. We had slightly higher p-values, but nonetheless had a trend toward significance in all these different outcomes. So really running up this ANCOVA model confirmed the results that we saw with the mixed effects model and the statistically significant findings we had shown earlier. And the subgroup analyses for the 300, 400, and 300 greater than 5 were similar. Differences were similar, p-values similar, but slightly higher. I'll again show you this pooled analysis, which was done using the ANCOVA model, because that was really the only way we could combine the data from study 7, study 20, and study 41. And again, we're here showing the highly statistically significant difference between Translarna treatment and placebo achieved over the 3 trials in over 700 boys with Duchenne muscular dystrophy. And again, those greater differences and still highly statistically significant in the 300 to 400 meter subgroup. So in summary, we're incredibly excited about these results and being able to show in a prospective clinical trial of a disease-modifying therapy for Duchenne muscular dystrophy, a statistically significant effect not only across the primary endpoint of 6-minute walk tests, but also in the other key outcome measures, including North Star and time function tests. And this study 41 really allows us to provide even more data to the totality and contribute to the totality of evidence showing benefit of Translarna, not only in clinical studies, but importantly in the real world over a longer period of time through our STRIDE registry, where we've been able to confirm a benefit of Translarna in delaying loss of ambulation and delaying loss of pulmonary function. These study results, we believe, certainly support the continued authorization of Translarna in Europe. They also continue to support the strong value proposition of Translarna for payers worldwide. And also, we believe, based on the strength of these data, we are in a position to discuss with the European Medicines Agencies a conversion from the conditional marketing authorization to the standard authorization in Europe. In addition, we believe the strength of the study 41 results allow us to initiate discussions with the FDA to align on a path for a potential NDA in the United States, which could hopefully finally allow us to have Translarna approved in the United States. So let me again by thanking you and thanking the boys, patients, families, and the whole DMD community for their partnership over these 2 decades. It's been quite a journey, as I shared earlier, with a lot of ups and some downs, but this is what pioneering is all about. And we're incredibly grateful for your partnership and look forward to continuing the next steps on this journey together with you all. Thank you.
We do have time for some questions from the audience. If there are any, you can go to either one of the 2 mics. Yeah, I can also ask while waiting for some. So you said that obviously going to start engaging, hopefully, with the FDA. Do you have a timeline for kind of, I think that's going to be, obviously one of the questions in mind for that pathway?
As you can imagine, we want to move as quickly as possible. FDA has a process for requesting a meeting. We will initiate that process as quickly as possible and then move forward to, if we're aligned on the NDA path, to do that as quickly as possible.
And with study 41, is there any plan with these boys to perform biopsies as part of the protocol?
It was not part of the protocol. This was a protocol that we designed as a confirmation study, really focusing on functional benefit. I think we're at a point with Translarna that the dystrophin levels, which are well known to not correlate well with functional benefit, wouldn't provide any additional information. Really, the key here was to establish clear functional benefit relative to placebo, which we did not only on the walk test, but on North Star and other tests, which really is such a significant finding that would outweigh any information we would gain from biopsies.
All right. Well, I think we can break for lunch a little bit early, so I'd like to thank you, Dr. Klein, for joining us and sharing that update. And thank you, everyone, for a great morning session.